Hi, everybody. So it's really cool to be invited here to give a talk. Last time I gave a talk here at Mount Sinai is when I pretty much just joined Cytobank, and that's when I met Adib and Jordi, and it's really cool to see just in the last year or so how successful you guys have become and really the amount of work that you've all put in here to really make the CyTOF and also get the data analysis going. So for those of you who don't know about Cytobank, I'm going to introduce to you the product as well as show what resources you have available here at Sinai and in general. Fluidigm and Cytobank have a partnership where Cytobank is the software platform for analyzing CyTOF data. So what is Cytobank? Cytobank is a platform for managing, analyzing, and sharing your single-cell cytometry data on the web. Any machine that generates FCS files from fluorescence to mass cytometry data, you can take that and drag and drop it onto your web browser and upload that data to Cytobank, where you have dedicated storage, compute, as well as gating and analytics software. From there, you can go and analyze your results and make new discoveries. And we have customers in industry, in research labs and academic institutions, as well as hospitals. And our goal is to allow researchers to focus on discovering the new biology and not worrying about where your files are and data loss and kind of the day-to-day grind with all of the data that's coming down the pipeline. For those of you new, it's entirely on the web, and so there's 3 different versions that you can access. There's our free version, our basic version online, which is Community Cytobank, and where you can just upload and share your data. A lot of our customers find this very useful for publication. For example, if you're going to submit a paper and you want your reviewer to look at it, or you're trying to satisfy making the data public as per NIH standards or from the journal itself, a lot of our customers will put data up there. There's also our premium version. Both our Premium Pro Enterprise versions are through our partnership with Fluidigm. The premium version is really good for individual licenses if you just want to try out our advanced tools such as plate annotation, Spade analysis, our Sunburst tool, as well as dose response curves, and VISNI, which is coming out next month. The third version is our professional enterprise version, which has all of the functionality from the earlier 2 versions, but also you have a dedicated administrator where you can have a lot more high touch on the site itself in terms of beta feature access, scalable computing, as well as administrator training. I've had, for example, one of our sites, we had a customer who wanted to run a really, really giant Spade run, and he contacted us, and we were able to spin up for him extra compute resources as well. So in some sense, it's actually having kind of a dedicated compute cluster at your fingertips. Just to touch on this, as Jordi mentioned, that there's also mountsinai.cytobank.org for those of you who are here internally, and probably contact Jordi or Deeb to get access. And that gives you all of the enterprise access that contains all of these 3 different platforms, the functionality within. I briefly want to talk about, in the news, there's a number of new publications that have some part of their publication hosted on Cytobank. And both of these, actually, the authors pre-publication contact us when they were submitting the paper, such that we could make the data available both for the reviewers, but also post-publishing. The one on the top is a paper which I think is pretty interesting from earlier this year, Charlotte Giesen from Bernd Bodenmiller's lab in Zurich, where they basically combined laser ablation for imaging with mass cytometry, where they were able to do single-cell segmentation and then run the data through our Spade algorithm. The second paper came out quite recently in Science Translational Medicine from the Nolan Lab. Brice Godelaire is an anesthesiologist at Stanford, and Gabby Fragiadakis is a microbiology and immunology graduate student at Stanford. And over, I would say, the last year and a half or so, they were monitoring patients post-surgery a few days to months afterwards, and they found specific biomarkers that correlated with clinical recovery. So the reason why I put some of these different publications, you'll notice if you see a Cytobank talk, we'll continue to put kind of the newest publications up that we've collaborated with. And is that there's a lot of interesting evolution within the domain, moving between different types of biological problems, from tissue discovery as well as from flow to CyTOF. But there's really been an evolution of Cytobank at the same time. And so how do we get here where we're the platform for analyzing high-dimensional cytometry data? Well, not many people realize this, but in fact that Cytobank was founded on a discussion where the three founders, Jonathan Irish, who was mentioned earlier, Nikesh, and Peter Kritzik, got together in a room and said, we have 550 samples from a fax caliber machine, and we just want to make a heat map, and we can't do it. And so basically in the Nolan lab, they started together with this conversation, which started the first Cytobank product. And over time, new tools and traditional tools like gating analysis have been put in. And as you can see on the bottom, kind of those technological tools as well have been improving over time, to the point where now Cytobank is a one-stop shop where you have a layered analysis as well as plate-based tools, our plate annotator, and sample annotation. CyTOF 1 and 2 really sparked a new group of algorithms, including clustering using Spade, and now VISNI, which is coming up next month in Cytobank. And we'll continue to push forward with new analysis tools to handle the data coming from new technologies moving forward. I want to emphasize, though, that it's not just the technology itself and the software itself, but really this is a biological problem. And for those of you who are already using the CyTOF, but are also interested in using the CyTOF for the first time, this is a quick kind of explanation, is that as the number of parameters have increased, so is our ability to measure distinct biological subsets in a sample. What you can see here on the top left is a graph of a few parameters, and as time has progressed, the number of parameters have increased, right, for those of you who have like an LSR2. And our ability to cover more different cell types, for example, within the immunological system has increased. However, in the last few years, there's been two major events that have happened. One is this increased depth in our ability to measure different proteins and parameters, but also breadth, both in fluorescence and mass cytometry, specifically within barcoding and plate-based tools, auto-samplers, for example. And the combination of these two has resulted in kind of an unprecedented amount of data, both from the depth perspective, but also the breadth, allowing you to cover a number of other types of cells, but extending towards cancer cells, solid tissue, apoptosis, cell cycle, et cetera, all in one sample run itself. And because of this, the shift from analysis is not necessarily waiting at the machine or running your samples, but more towards data interpretation and experimental design. And to accommodate this, one of the things I'm going to talk about first, which often we kind of put at the end of our talks, is the visualizations and advanced data analysis that Cytobank provides for demystifying this biological complexity. Three of the tools that I'm going to talk about today briefly include our Spade analysis tool, the population Sunburst, as well as VISNI. Earlier talks that you saw today covered Spade and VISNI. Primarily today, I'm going to focus on more of the analysis tools themselves because you have a lot of wonderful biological talks, so I don't have to really go into that too much. So the first one I'm going to talk about is Spade. So with the original CyTOF paper in Science in 2011, Spade also debuted. And the general, for those of you who don't know about Spade and usually do kind of manual analysis of your data, Spade kind of takes it one step further and auto-finds a number of populations within your sample subset. It does that by first downsampling to preserve rare populations, and then clustering all of your data in your n-dimensional space, which you usually define. So for example, if you're interested in all of your T-cell subsets, you can put your T-cell markers into Spade and cluster based on that. However, at this point, if you have your data in high-dimensional space, Spade then creates this minimum spanning tree visualization, which allows you to lay this flat on a page, so you're able to look at the different clusters as well. You can think of it as a heat map, which also has a lot of biological information built into it. This example from the original Science paper in 2011 really depicts this, where within one CyTOF run and one Spade run, you're able to capture the entire hematopoietic continuum. Just in terms of details, each individual node is a cluster of cells. Traditionally, you might manually gate some of these. And then each bubble is something that a biologist has gone through and designated as a population of interest. And so you can do this directly in your web browser and Cytobank. The color that you color each node by is the expression level of a marker. So in this case, for example, this tree is colored by CD45. And then the sides of each node represents the number of cells itself. Similar to as I was saying before about how you can think of this similar to heat maps, is that when you run Spade, you actually combine all of your samples together. So it could be controls and patients, or in this case, a basal sample as well as other samples that have been stimulated. For example, here's the basal levels on the left of STAT5, and on the right you have IL-7 treated samples. And what you can see is that within Cytobank, you can take these two, lay them on top of each other, and look at the fold change of your STAT5 signaling within the context of all the different cell subset populations. And you can do this across many different stimulation conditions, and even, for example, drug response. For those of you who are interested in just getting started, on Fluidigm Cytobank, we have example data sets, as well as kind of step-by-step. At the very top left, you can see this basically a unbubbled tree, and then we go through the steps of annotating those cell subsets as well. In addition, on Fluidigm Cytobank, for the same experiment, we have an example gating strategy for the experiment as well, which, as you can tell, as you're getting to more and more channels, your gating strategies start to become more complex. And this really leads into the next visualization tool that I want to bring up is the Sunburst, the population Sunburst, which we released earlier this year. So the population Sunburst, for those of you who already have experiments on Cytobank, you can go to the gating page and look at this right now. Basically, underneath your gating strategy, you can see that usually you have this population hierarchy right here on the left-hand side. And you have things like percent of parent and total event count. And as you create your population, Cytobank calculates these event counts. But the thing is, it's very difficult to go through and see what the relative fraction of some of your cells are within this entire table. And so what we've done is we've put a visualization in, which allows you to view it all in one place. Specifically, what you can see here, the very center is the top of your population hierarchy. And as you drill down to your T cell subsets, et cetera, these get split further and further down. One of the things is that one of the things that's very useful about the population Sunburst is that it's interactive. Specifically, you can interact by moving to different higher populations. You can mouse over to see the event count information for any of these individual wedges, which represent a population. And each ring represents all populations with the same parent population. If you click on, for example, your T cells, it'll zoom in so you can look at the Sunburst for just your T cell subsets and look at the relative fraction of those cells. The third visualization tool that I'm going to talk about, which we're debuting next month and releasing to our enterprise sites, is Visni. So you heard a little bit about it earlier today, and you're going to probably see some plots later this afternoon as well. But Visni is a dimensionality reduction tool. So if you've heard of, for example, like PCA or local linear embedding or isomap, the general idea for all these methods is that you have your data in high dimensional space, and you just want to look at it in 2 dimensions or just a couple dimensions so you can really see what are those really distinguishing features of your sample. This has applications for just identifying known populations, but as you saw earlier within Aaron's talk, that you can also start to identify populations in patients within disease. And so what we've noticed for a lot of our customers who have been beta testing Visni are that they've been looking over, for example, temporally for patients or thinking about different control versus patient samples. This original figure, as Erin pointed out earlier today, is this patient up here at diagnosis and then at relapse. And those events that are distinguishing of relapse are high in very stem-like markers for stem cell markers such as CD34 positive as well. The way you'll be able to use Visney and Cytobank is pretty simple. You just have to put in, you just take your data that's already on Cytobank, put how many events you want to run it on, and then afterwards you can view it in 2 dimensions. And the idea is that I just want to emphasize why dimensionality reduction is really useful. Even in 3 dimensions, if I look at this data right here, it's really hard to distinguish all of the different populations that I want to kind of go in and identify. And what Visney does is it really allows you to put it flat on the page such that you can go and be confident and annotate those specific cell subsets as well. And proof of principle, one of the things that we've done is running it on canonical data sets that we already have on Fluidigm Cytobank. For example, this is a 26 marker data set that Jonathan Irish collected a while back, so for each of these plots, the x-axis is one of the Visney dimensions. The y-axis is the second Visney dimension. And we've colored every single point by a particular marker of interest. So on the top left, you'll see that those cells on the bottom right are CD19 positive. On the left, you'll see those are your CD33 positive monocytes. And on the bottom, you can look at your CD4 positive T cells. And right here at the top, those 2 little populations, those are actually 123 positive cells as well. So just to briefly summarize, what I've talked about primarily so far are the tools that a lot of people come to us first to use because we're kind of the one place that allows it to be done quite easily within one platform. For example, customers will come to us for clustering tools such as Spade and Visney and our Sunburst visualizations and gating analysis. But the rest of this talk, for the rest of this talk, I want to talk a little bit more about all of the other things that make this possible within the platform, which include the raw data processing, as well as summary analysis and annotation. A lot of different tools that we have, for example, dose response or plate annotation or our statistics export. The first thing I'm going to talk about is pre-processing and gating briefly. Pre-Cytobank, we have a few tools that we have hosted on our site, for example, our FCS concatenation tool, as well as there's other steps pre-processing before you do your analysis, such as bead-based normalization, and for some of you as well who are starting to get into it, debar coding. I believe right now bead-based normalization that's built into the CyTOF 2 software. We also host the original software published, I think it was the year before last. Within Cytobank, we also provide kind of all the basic tools for manual analysis, which include automatic compensation for fluorescence flow cytometry, as well as gating analysis on our platform as well. And that's an example of gating on your Visney plot within the Cytobank platform. The second thing I want to talk about, which is very dear to my heart as well as I think the entire Cytobank team, is storage and shared annotations and really reproducible research. We're at a point in time where data is just exploding, right? If you have, if some of you have the new iPhone, it's collecting how many steps you're taking all the time as you're walking around. And the sheer amount of data has just exploded in the last couple of years. And scientific data is definitely part of that group. Right now, on average, scientific data is growing at approximately 30% per year. And the problem with this is that there's a number of new studies, for example, this paper from Vines last year, that we're losing the data over time. They basically went through a series of publications and tried to determine whether or not the data was accessible. Currently, we're decreasing at approximately a rate of 17% per year. And even for those that we can still find some of the original information, the results are not reproducible. And more than 50% of non-reproducible results were either due to unavailable data or unavailable software. And so the key is centralizing the data, analyses and all of the meta information in one place. And so this kind of comes back to the original purpose of Cytobank, which was for data storage, annotation, and sharing, where you can upload your protocols, you can put your plate annotation and information. A lot of this, if you're using an auto sampler, for example, on the CyTOF or the LSR2, this automatically comes into Cytobank, and you don't have to put that information in. And really kind of looking at the different layers of analysis. At the bottom, you have the raw data down here, and then you have your 2D plots, but also your summary plots and statistics. And the idea is if you ever change a gate or something in a compensation matrix, that this gets reflected in your final figure, and that you don't have to worry about all of those steps in between. The other thing I wanted to mention that not many people know about in Cytobank is our tools for templating analysis. Let's say you're running a cohort study over many different time periods, and you're running the same panel over and over again. You can copy your gates and your annotations from experiment to experiment over and over again so that you can just open it up, tweak it a little bit, and then move on. In addition, you can copy whole experiments between different within a Cytobank, but you can even import and export your gates, your annotations, and your experiments and move it to a different Cytobank. For example, for our customers who have a lot of data on our free Cytobank and want to try running Spade, you can either download and transfer your data over yourself, or we can actually transfer your entire experiment over as well. Sharing is also a really important part of the Cytobank platform, and there's 5 different ways to share. At the kind of lowest level here is that by default, when you upload your data to Cytobank, everything is private to yourself, and you can do all of your analysis, your gates, independently. However, what we've noticed is that most people like to do full access, where you can just share with a collaborator, and you can work on your gates and Spade trees together, even on your iPad. The next level is limited project access. I like to think of this as the PI block. Your PI wants to look at your data, but you don't want them to touch your gates or anything that you've put a ton of effort into. And so project access will allow them to view your figures, but not touch something underneath, and you can regulate that access. It's also really good for reviewers if you're submitting to a journal and you want them to, if you add them to a project to view the data. Public experiments are institution-wide. So, for example, if you had an experiment and you've published it and you just want to show it to Mount Sinai, for example, you can make it public with one click on Cytobank. You can also remove it from being public if you want as well. On Community right now, I think we have a couple hundred experiments that are publicly available post-publication. The last one I want to talk about is Cytobank reports. Some labs have worked with us to put up whole landing pages as well as reports. So if you want people to look at your data and you don't want them to require a Cytobank login, some of our consulting efforts have been to put up data associated with publications and link to the original experiments on Cytobank as well. And so that's basically for people who want to submit a Cytobank report along with their publication and link to it as well. Okay, so the last thing I'm going to talk about is collaboration. And this is really, there's been this evolution because earlier I was talking about collaborating with visualizations, collaborating through sharing, but the real emphasis of this is that even though you're collaborating, every person that you're collaborating has a different role in your team. Specifically, you might have individuals who are working at the flow core, or you have those who are designing the biological experiment. You might have a statistician and a computational collaborator, and you might have a clinician. And all of these people are part of a team. One example of a project that I'm working on right now is an ovarian cancer project, which has every single one of those roles filled right now. And everyone wants to come to it at a different level of the data. And the idea is that if you have it all centralized in one place with the layered analyses, you can come in at any point and you can see, for example, if someone's generated a figure, you can take a look, copy it, play with it. Or you can also get to the raw data files, export that transformed data, and put it into some statistical tool like R. What we've noticed as well is that some of our customers are actually taking data out of Cytobank, processing it, and putting it back in for sharing as well. And one initiative, for example, is there's a few people, for example, Nima Agapur, who's in the Nolan lab, who has built an auto-gating tool. And what he's done is, and I think it's not in submission yet, but soon, I think he presented it this year at CyTO, where to help with sorting, once you've done your large CyTOF experiment and you want to sort a specific type of cells, you can actually develop a gating strategy. And he writes those to gating ML using R, and then I import those into Cytobank onto existing data sets. And so a lot of these collaborations, oh yeah, this is work with Aaron as well. He's nodding, he's like, I've seen those gates. But what's really interesting is that there's a number of groups of collaborators that are not only working within Cytobank, but taking data out and putting it back in. The last thing, though, kind of the taking data out, one of the important things that we have in Cytobank is statistics. And you can visualize your statistics in the heat map form or other forms, Pearson correlation, standard deviation, percent and gate, things like that. But if you're working with a statistician and you want to pull that data out as well from Spade or from your gated analyses, you can actually use our export statistics tool to put the statistics plus columns with annotations out into programs like Excel and R. The reason why the annotations are extremely important is that if you're working in software such as like Spotfire, Tableau, or Jump, those annotations become very powerful when you pull it into an external analysis tool. So in terms of integration, one of the things I wanted to mention is that I talked a bit about the analysis, the raw data processing, as well as kind of the summary statistics and annotation. I didn't have time to go too much into our dose response or our plate annotator tool, but we have a number of other tools within Cytobank which are particularly useful for large studies if you're running large-scale assays. However, just focusing, for example, on what we're releasing next month, which is VISNI, dimensionality reduction, we've been very careful to integrate it throughout the entire platform. So any new tool that we put in currently leverages all of the different parts. For example, you may first go through and do some general analysis or plate annotation before putting your data into VISNI. However, after that, you can take your VISNI FCS file now that we've generated, you can download it, you can gate on it and look at the Sunburst, you can actually run Spade on your VISNI channels, you can gate your populations and look at it in heat map form. And so we're not innovating in terms of trying to think about all the different combinations. We're relying on you guys to figure out new ways to combine all of the tools that we've put into the Cytobank platform. And we've already found a lot of really interesting questions from just the folks that have been beta testing our software thus far. For those of you interested in getting started with Cytobank, there's our tutorials. So we have our YouTube channel. So if you just Google Cytobank YouTube, you'll see a number of videos showing how to get started with Cytobank. We also have our quick guides on our blog, which links to not only data sets that are available on Cytobank, but step-by-step, like how do I gate in Cytobank? How do I run Spade? Things like that. We also have tutorials on PBMCs for fluorescence and mass cytometry data. If you're interested in getting help online and just reading about Cytobank, we have our support portal, which is basically a giant FAQ with any question you might have. It has a lot of answers within there as well as the Fluidigm Cytobank link there, which has more mass cytometry relevant examples. In addition, our support team, Jeff's over there, who's part of our support team. Some of you might have interacted with him before. We have a weekly webinar, which Jeff often gives, and so does Chris, where we do it live every week. And basically, we have interchanging webinars for introductory analysis and then advanced analysis, for example, Spade, and sometimes we touch on the plate annotator tool. And we'll also follow up and bring things like VISNI in as well. Afterwards, you can also ask questions and talk to other people, or I think you can just ask questions to Jeff. I don't know if you can talk to other people after the webinar is over. But in addition, if you email helpdesk@cytobank.org, we usually reply pretty quickly, as well as we have a chat room, which you can pop in A story I like to tell, and I think some of you might have heard it before, is that I remember I have gotten texts at 3 a.m. from our sysadmin who says, there's someone in the chat room, they have questions about Cytobank, can you answer it? And so although the person who might be in the chat room may not be able to help you immediately, they'll make sure to get you a response quickly. And the other thing I have is that if you do come into the chat room, please write your full name, because we've had people who put part of a nickname in there and we can't find them later. So the last thing I wanted to touch on in terms of getting started, as well as kind of advanced support, is that something that some of you may not know is that we have analytical services and on-site training available as well. Some of the projects that you see, as well as ones that are coming down the pipeline, like the ovarian cancer project I talked about, are things that we do consulting on for helping people understand and do analysis. So for those of you interested in the webinar, our next one, well, there's one today, but hopefully you're here for Brent's awesome talk, is the Introduction to Advanced Analysis, which will be next Wednesday at 1 p.m. Eastern Time. And then also some of our folks are going to be attending different conferences in the near future. Nikesh, our CEO, will be at the French Society for Immunology next week. And then there's also the Autumn Immunology Conference as well, which I think Jeff is going to be attending. With that, I'm going to conclude. This is a picture from the beginning of our year at our Cytobank retreat. And since then, when I first joined the company about 1.5 years ago, we were at 7 people. And now, at the time of this, it was about 19, and we've continued to grow since then. And so I just want to thank you all for your time and welcome any questions.
This is a fantastic tool and much needed in the community.
Thanks.
So I'm always really impressed. I haven't used it myself a lot, and one of the problems that we encountered previously when we clustered data, in particular between different experiments, even if it was the same donor, is that in particular unsupervised clustering doesn't find the same clusters again. How good are you in comparing data sets and finding the same clusters?
Yeah, that's a really good question. And part of it's really interesting because some of it has to do with like your sample variation and the fact that there's a reason why gating has existed for so long because something that is statistically significant may not necessarily be biologically significant. We have a few tips and tricks that we usually do within Spade analysis itself. Which include, if you're interested in rare populations, we encourage you to increase the number of clusters because that creates kind of a tree that's much more reproducible in terms of the structure of the tree. So you're able to kind of find those kind of more blurry lines between populations. What we've found really interesting, actually, is that running VISNI first on your data and then running Spade on those VISNI dimensions. There's a number of other tools I think that have come out that are being published that do kind of similar things, really allow you to find clearer, distinct populations that you can then further investigate and run your tools a second time on to kind of really dig into those subcellular subsets. I think it's still it's a partially solved problem because there's so much biological variation. And so I think often what you do is your first Spade tree is not necessarily the one that you're going to do for publication. You run it first, and then you find out which samples are interesting. You'll also determine which of your markers stained really well versus those that may not have. Spade is very good at finding debris and just kind of those kinds of items. And so you can filter that out and rerun again. And so part of it is that it's really for discovery of those interesting cell subtypes. And to recapitulate them over and over again, you have to really think about the data and make sure that what you're getting is real, which having the Spade tree plus the 2D plot on the side helps aid with. But I would say that for your samples, part of it is potentially looking at the markers that you're running on, and first picking those that you know are very distinct and will help you find those subsets first, and then investigating further. I would also say that when we release VISNI next month, that it's really helped some people tease out some of their subsets, both with VISNI alone, but also the combination of the two.
Thanks. And I have a second question,
Sure.
completely different. One of the things that industry has qualms with is putting their data up there. Is there actually a way of using all of this offline, or you have to have your data in the system in order to analyze them?
So everything is currently hosted on the cloud. And what's interesting, though, is that that hasn't necessarily stopped us from having basically a number of our customers are pharmaceutical companies. And so we've been using them kind of as the standard to show that our Cytobank platform is secure. We're also currently moving to AWS, which has extra security, and we're at FSMA moderate right now. So for those of you who try to keep your data up to that level of secure, that's what we have right now. And so it's becoming less and less of a problem, especially some of the consulting work we're doing with some pharmaceutical companies. They actually ask us to transfer our data to a cloud-based storage so that they can pull it as well. So I think it's becoming more and more a standard practice. I would say 5 years ago it was not.
Can you import into Spade non-cytometry data to do analysis that don't have marker numbers, like sequence data, for example, to look for patterns?
Sure. So yes and no. So actually last year there was a publication in Cell Stem Cell which did a I believe it was a RT-PCR or qPCR data set where they ran Spade on that. And ultimately an FCS file is a matrix. And so what I've done in the past as proof of principle, and some people are doing this as well, to get it into the Cytobank platform, because currently we read FCS files, is that you can actually convert your data matrix into an FCS file and upload it to Cytobank. One thing, I don't have it here, it's a little bit silly, but I have this one slide where we basically ran Spade on a friend's face. And so you can actually convert imaging data into an FCS file and run Spade on it. And when we release next month, in addition to Visney, we're putting out these Z-colored plots where it's coloring by a third channel, which also helps with that type of analysis. So I would say that it's possible, but you have to convert your file over. And there are definitely tools for that, one of which is available on Gene Pattern. Josten Spindlin up in Vancouver integrated that there. So you can just upload a matrix and it'll convert it into an FCS file.
Hi, so I run a clinical lab, and the question that I'm wondering about is, do you have any app on the client side that can actually strip patient data from FCS files? So I'd be very interested in using your tools, but in no way would I upload data that has patient identifiers in it.
And we request that you do not do that.
So in other words, there is no app that I can pre-strip out that data out of FCS files.
We do have, and I'm trying to think about some people at Stanford and Harvard, they strip out the patient identifying information first, and they'll map it to some type of annotation ID instead, and then upload the data to Cytobank. It's true, currently we don't have kind of the HIPAA certification where you can put that patient identifying information on Cytobank, but the FCS files themselves, you can strip a lot of that information out. We don't have a tool currently, but if you email me, we can try to find someone to set you up with that. Thanks. Thanks.